technical difficulties getting started. Tonight I'm going to begin an ongoing series of talks on cancer, and the title of this talk is Cancer, a Metabolic Disorder. So the question that we are going to ask tonight, is cancer more of a metabolic disorder than a genetic disease? A revolution is happening in the field of oncology, and quietly behind the scenes you may not have heard about it, or perhaps you have and you're well versed in this. An enormous revolution is occurring in a rethinking about cancer that is very, very exciting. So a few times during my talk tonight, I will invite any listener to schedule a personalized health assessment for adjunctive cancer care for you or your loved one at the number that's listed here on this slide. You can call 202-237-7000, and extension 152 will get you to the Functional Medicine Genomics Department at NIA. So is cancer a metabolic disorder more than a genetic disease? Because for most of the history of oncology treatment, it has been considered to be a genetic disorder. And I have been working with my patients and using this kind of model for years now, and we continue to test for the genetic basis of cancer, like BRCA genes, to determine my patients' inherited vulnerabilities. That still is a very useful model. And increasingly, oncologists are getting very good at testing cancer cells to determine their unique genetic vulnerabilities to the various chemotherapies. But cancer may be more of a metabolic problem than a genetic one. Now, what I'm going to present tonight is a model, and models change as science advances. And the great George Edward Pelham Box, the famous British statistician, is quoted as saying, all models are essentially wrong, but some are useful. So models change, and I'm going to discuss this metabolic model tonight, but who knows what's going to happen in the future? Who knows what kind of new models will emerge that are more useful than even what I'm going to talk about tonight. And you've probably heard of Stephen Hawking, the famous astrophysicist, who has written a book called The Grand Design, Chapter 3, What is Reality? And reality is the models that we perceive in life. And it can be models for interaction with our family, or models of how the universe works, or models of what keeps us healthy. But Hawking defines a good model as being elegant, contains few arbitrary or adjustable elements. It agrees with and explains all existing observations, and it makes detailed predictions about future observations that can disprove or falsify the model if they are not borne out. So the paradigm shift I'm talking about in this model of what cancer is towards a metabolic model and away from the genetic disease model. The genetic disease model is based on mutations, somatic mutations in the oncogenes and tumor suppressor genes, that is, genes that suppress tumor formation. They mutate and then cancer is not suppressed anymore. And this has certainly enjoyed success. But the model that cancer is also a mitochondrial metabolic disease promises to greatly enhance that success. And this is so exciting. And it's right up the alley of those of us that practice functional medicine, because metabolism, the immunity, the allergies, the nutritional deficiencies, the toxicities is what we work with, getting to the root causes of why we get sick, why we age, why we die. So the model that cancer is a mitochondrial metabolic disease segues into rational, obvious treatment strategies, which then can generate an inhospitable physiological environment, making it far less likely that cancer cells can thrive. So I tell everyone now, I don't care what kind of treatment for cancer that you utilize, surgery, radiation, chemotherapy, integrative strategies, herbs, you name it, whatever you do, whatever you can afford, whatever seems right for you, make sure you address this metabolic issue. And whatever it is that you do, you almost would have to do better. So the model that cancer is a mitochondrial metabolic disease, from that angle, segues into treatments which satisfy the 4 criteria defining the value of all treatments. What 4 criteria define what a good treatment actually is? A good treatment is reasonably safe, safety first. A good treatment is reasonably effective. A good treatment is reasonably simple, easy to do, and a good treatment is reasonably inexpensive. Safety, efficacy, simplicity, and expense are the 4 criteria that define what a good treatment is. And the treatments from the metabolic angle fit those 4 criteria. Now, what's very interesting to me in medicine, particularly coming from the addictions treatment, is that we're always prescribing things for patients, and we're rarely proscribing. So I have for some time now been joking with my colleagues, my family, with my patients, that I have a proscription pad. So if an alcoholic person comes into me for treatment, I give them a proscription for alcohol or tobacco, a prescription for tobacco, because we're heavily laden on the side of prescribing, doing things to people. You know, here's drugs, here's a treatment, and we don't do enough proscribing or supporting patients in proscribing those things that are obviously harmful, like alcohol and tobacco. And this is the definition of proscribe: forbidding, denouncing, etc. So it turns out that from this metabolic angle, this is just the first of several webinars I plan to do, but we need to start at this one place, and then we will branch out into other dimensions of this metabolic cause of cancer, this metabolic approach. So it turns out that cancer cells are sugar addicts. It's plain and simple. They're carboholics. They depend exclusively on sugar for their survival. Now, what is regular table sugar? It's a disaccharide. It's actually 2 sugars, glucose and fructose. Now, cancer's primary food is glucose. A lot of different sugars will eventually convert into glucose. That's the food that keeps it alive. But interestingly, totally unexpectedly, are studies suggesting now that fructose is responsible or assisting in metastasis. That is, cancer cells break free of their original site of growth and migrate to some other part of the body. And it's generally accepted that 90% of the cause of death from cancer is due to metastasis. So this is a double whammy. This stuff, glucose, feeds the cancer, and the fructose helps cancer metastasize. So cancer is sugar is a very, very bad thing. So cancer cells are sugar addicts. I'll explain this later. They're carboholics. The more undifferentiated, the more primitive a cancer cell becomes, the more they derive their energy exclusively from sugars by a process called anaerobic glycolysis. Now, I apologize. There's a few technical terms that I'm going to introduce tonight, and I limit them wherever I can, but these terms are just so definitive of what the issue actually is that it's hard to leave them out of the picture. So glycolysis is the breaking down, lysis of sugar, and it's an anaerobic process. That means there's no oxygen involved in that breaking down of sugar. And so that what is technical shouldn't be over anybody's head, the NBC News has, this is just one of the media presentations that I bring into this discussion. And the news is supposed to provide information that is at a relatively simplistic discussion so that as many people can engage in understanding what they're talking about. So even NBC News, even the media is presenting information on this. And the presentation was, here's how sugar might fuel growth of cancer, January 2016. And remember, starches, complex carbohydrates, rapidly are converted into sugar. So I'm not just talking about sugar, I'm talking about all carbohydrates, all starches. So if you take one thing away from tonight, cancer cells are sugar addicted. And the more we can address blood sugar levels for any victim of cancer, the better they're going to do, to the extent that this metabolic model carries a lot of weight in determining the outcomes for individuals with cancer. So if you take one thing away, cancer cells are carboholics, and anything that can be done to lower blood sugar levels can only help an individual who is suffering from cancer. So here's a few things that we're utilizing. We've got some wonderful possible interventions on the drawing board. So diabetic medication is showing great promise. I have a reference at the end of this talk on glucophage. Insulin potentiation therapy. Insulin is provided to lower blood sugar levels, and then whatever is done with a lower blood sugar level, cancer cells are extremely vulnerable. A Paleolithic diet, paleo diet, with absolute sugar, carbohydrate restriction, starches, most fruit. Now, fasting has often been a holistic medicine healing modality for all kinds of problems, including cancer. And you'll hear stories, you'll have testimonials about patients that used fasting as a way of treating their cancer, and they did very well. Now we know why. Now we know that by restricting carbohydrates, the cancer cells were starved. Exercise, obviously, is going to burn off sugar. Why do patients in recovery from cancer who engage in exercise programs, why do they do better? Now we know why. And there's some other newer drugs like sodium glucose transport 1 inhibitors, which should be more promising even than glucophage. And there are natural analogs to the SGLT1 inhibitors from apple tree bark and leaves that we're looking into, which should be totally harmless and without side effects. Now, in case people are coming in late, here is the information to contact us. Tonight we're talking about cancer as a metabolic disorder. So personalized health assessments can be scheduled for this adjunctive cancer care for you or your loved ones by calling this number, extension 152. All right, so where is it that this metabolic problem is happening? So if you look at a cell, this is a diagram of cell, and I thank Wikipedia for this, that I don't have to become artistic and draw my own cell. And these are the different components of a typical animal cell, from the nucleus to the ribosomes to different vesicles, the rough endoplasmic reticulum, and so forth. And if you go down to number 9 here, you see these little guys. They're called mitochondria. And this is the outside of a mitochondria. But they sliced it off in half so you could see the cross-section of the mitochondria. That's where the action is. And as I mentioned earlier, we always thought, and there is evidence, of course, that the DNA damage in the nucleus of the cell or on the cell membrane at receptor sites or in various places of the cytoplasm inside the cell, most of the research, most of the energy in oncology, whether integrative, whether conventional, has gone into that discussion. And I think we still should look at that. We still need to do genetic testing to find the genetic vulnerabilities. We still need to find the genetic vulnerabilities of cancer cells and tailor the treatments according to what we find. But the metabolic problem is right here in the mitochondria, which generally has not been the subject of investigation in many areas. And the mitochondria explains so much that we never knew had such a major impact on cellular physiology. Now this is the little mitochondria, a cross-section of it here. And what part of the mitochondria is involved in this metabolic issue? It's right here on the inner boundary membrane, right here. So you see this membrane that almost like a brain, you know, with the way it goes in and out with these little indentations, and that's where literally the high energy activity of the cell is taking place. See, so here's this cell membrane protecting the cell. Here's the cytoplasm of the cell. Here's an outer layer to protect the mitochondria. And the high energy thermonuclear activity is happening inside the mitochondria on the cell membrane. So I'll give you a little hint about future talks. Where is it likely that viruses, radiation, or toxins are going to damage the cell? Well, it could damage all kinds of places. It could damage the cell membrane, but it is far more likely that the damage would occur where this extremely high energy of ATP production, the fuel that runs life, it is far more likely that the damage is going to happen on this inner boundary membrane. And so what we can do is just look at this membrane. See, here's the outer membrane of the mitochondria, a schematic, and here's the inner membrane. So we have these cytochrome systems that take the NADH. Again, I'm sorry about technical terms. You can think of NADH as activated vitamin B3, niacin. And this activated NADH that's burned in the citric acid cycle. Now, all my patients know what a citric acid cycle is because I test it in virtually all of my patients, because we're looking for the source of fatigue in so many patients. Is the fuel being burned, the fats, the carbohydrates, and proteins, is it being burned in the Krebs cycle or citric acid cycle? And if it is being burned, then these intermediaries are made like NADH and FAD and so forth. And they interact on this inner mitochondrial membrane. This is where the explosive, high energy stuff is happening in the cell. Everything else is quiescent compared to the mitochondrial membrane. This is where oxygen is just burning, exploding. And this is the place, again, where damage can occur. And I suppose most people know that viruses, toxins, and radiation is carcinogenic, causes cancer. But did you ever wonder, how do these things cause cancer? And we have always believed it causes mutations. So think of all the scary science fiction movies. Think of Spider-Man. He gets bit by a radioactive spider that causes mutations, and he has superpowers of Spider-Man. Well, it turns out that the damage from a toxin or radiation is far more likely to occur where there is an exchange of extremely high energy, where there is a fire happening, literally oxidation happening. And it's happening on this inner membrane of the mitochondria. Right? All right. So all of my patients get a diagram of the Krebs cycle, which demonstrates how fats, carbohydrates, and proteins, the three main energy-producing components of food, how they come in through a common pathway into the mitochondria to be burned to make NADH, and the NADH and the FAD then goes to the mitochondrial membrane where it is converted into ATP. And of course, we're interested in this because ATP is the fuel that runs life. If we can't make ATP, well, if there's some interruption in the Krebs cycle or this mitochondrial membrane, then we're going to be fatigued. We can't make hormones, neurotransmitters. Our heart could be jeopardized. And the brain also, because the heart and the brain are by far the two highest energy consumers in the body. So the production of ATP is very, very important. All right, so all cells, undifferentiated cancer cells and differentiated normal cells. That's another term. Differentiated means highly evolved. The more highly evolved anything is in nature, the more capable it is of burning this explosive fuel of oxygen. Most of Earth's history was anaerobic. There was no oxygen. And it was a time, life was there, but it was a time of single cell, very primitive organisms. When oxygen became available and this explosive fuel became available, that allowed for multicellular organisms to evolve, and the last 600 or 700 million years out of 4 billion years of Earth's history has involved an interaction between these undifferentiated primitive cells like bacteria and differentiated mammalian animal cells that make up the 200 trillion cells in our body. So all cells, whether they're undifferentiated or differentiated, primitive or highly evolved, first of all, derive 2 ATPs from what's called glycolysis. So that's this step here. Sugar is made into pyruvate, and that derives a net 2 ATP for energy. And that is called glycolysis. Now, if the mitochondria are still working, if the citric acid cycle is working, and remember, this is not the most vulnerable place in the mitochondria. It is the inner cell membrane that is. But if the mitochondria is still working, 2 more ATPs can be synthesized in that process, and that's called substrate level phosphorylation. Just forget the term, but the main thing is that the primitive derivation of ATP that all cells are capable of involves the conversion of carbohydrates into pyruvate and the 2 more ATPs that can be obtained through the Krebs cycle or citric acid cycle. This process is anaerobic, does not require oxygen. Now, all cells, undifferentiated or differentiated, can derive an extra 2 ATPs from the Krebs cycle. So we went through that. So the total is only 4 ATPs in a completely undifferentiated cell. In other words, if a cell is completely undifferentiated, completely primitive, the most it can get is 4 ATPs from carbohydrates, and the damage to the mitochondria may be so severe that even the citric acid cycle doesn't work. And that's a fully undifferentiated cancer, fully primitive. The only way it can get energy is from sugar to derive two ATPs and the synthesis of pyruvate. So this is when not only is the Krebs cycle, not only is the cell membrane, that's the first thing that goes, because the cell membrane of the inner cell membrane of the mitochondria is where this explosive energy is happening. That is wiped out by heavy metals or viruses or radiation. So that's the first thing that goes. But the mitochondria is so vulnerable that even the citric acid cycle can go. And if that happens, then only 2 ATPs can be derived from food by a cancer cell. It can only take sugar to pyruvate. That's it. Now, in a normal cell, all that's possible too. But also in a normal cell, fats and proteins and carbohydrates come into the mitochondria, so all 4 ATPs, no problem. But these byproducts of the Krebs cycle, NADH, you see them coming off here, and FAD, these go into the mitochondrial membrane and can produce up to 30 ATPs. This is called oxidative phosphorylation. It is extremely evolved, highly efficient, controlled thermonuclear energy production. Let me go back again to make it clear. So when this inner mitochondrial membrane is damaged, that oxidative phosphorylation disappears completely. All that a cancer cell can do is make pyruvate, 2 ATPs, get 2 more ATPs out of the citric acid cycle. Further damage, because all this explosive energy is going to damage everything in the mitochondria when damage occurs, and the citric acid cycle can be wiped out as well. So even that's gone. So a totally undifferentiated primitive cell cannot do this. Cannot do this. It gets its energy only from glucose to pyruvate, 2 ATPs. A normal cell can take fats, proteins, carbs, run all this through the Krebs cycle, make ATP there, but then take these byproducts, put them into the mitochondrial cell membrane, and kaboom, 30 ATPs. So a normal cell can make all of this energy. A cancer cell cannot. Cancer cell is limited to only 2 ATPs if it is totally undifferentiated. The more damaged the mitochondria, the more the cells become totally undifferentiated, the less that substrate level phosphorylation works, even the meager 2 ATPs in the Krebs cycle, and the more totally the cancer cell depends exclusively on sugar to maintain its energy requirements. In other words, a very undifferentiated cell cannot not even get energy from fat or protein anymore, only from sugar. It is addicted to sugar, and here's why. A cancer cell could be 20 to 100 times more dependent on sugar than a normal cell. And the reason for that is the energy requirements of all cells, cancer or normal cells, is relatively constant. See, to be alive, all cells must maintain a certain level of energy production, which is in a fairly narrow range. If that energy production falls below that point, the cell dies. So I won't get into the chemistry and physics of it, but whether you're a cancer cell or a normal cell, minus 56 kilojoules of potential energy must be maintained. The energy requirements is the same. But in this diagram, what we see here is a differentiated normal cell, and then the cell becomes, the mitochondrial membrane becomes damaged, some, but it's still pretty functional, not cancer yet. This is like a grade 3 pap smear. Or a polyp in the colon that's beginning to look weird. Now it's definitely getting weird, and then it becomes fully cancerous. And this is a progression from no mitochondrial damage or relatively little to full mitochondrial damage. So in the healthy cell, at least 85% of its energy comes from oxidative phosphorylation, from the production of ATP through oxidation of fats, carbohydrates, and protein. And a normal cell gets 12%, 10% through anaerobic glycolysis. But as mitochondria become progressively damaged, that anaerobic glycolysis, the energy requirement is more and more and more dependent upon anaerobic glycolysis, which can only get 2 ATPs. And as this progression to cancer occurs over time, the ability to derive energy from oxidative phosphorylation goes down, down, down because of the mitochondrial damage. Now, we can devise treatment strategies to take advantage of this and attempt to get a cancer cell sober from its sugar addiction by lowering blood sugar levels. So that's what this diagram is intended to suggest. This is, and an addict, a severe addict, really has 2 choices. They can either be rehabilitated, and this also gives some hint about future webinars. How do we rehabilitate our cancer cells and repair the mitochondria and bring them back to normalcy, or deprive it so much of sugar, which is its only food source, that it falls below the necessary energy requirement for it to stay alive. Now, there are a lot of other components to this, which I'll talk about in future webinars, such as the process by which a cancer cell or a cell that's becoming a cancer cell decides that it's time to die. It's called apoptosis. And the mitochondria, of course, are heavily involved in this apoptosis decision. And if the mitochondria are damaged, then the cell could be on its way to cancer, and it basically loses that signal that it's time for it to die. It's time for it to clear out. And it goes on to become a raging cancer cell. So we need to give these cancer cells a choice. Either start learning how to metabolize food normally again, heal your mitochondria, or die. And that's the choice we give addicts. A severe addict basically has two choices. You can rehabilitate and get sober, or you die. Heroin deaths now are greater than auto accidents and gunshot wounds combined with this heroin epidemic. It's tens of thousands of deaths every year. So we try to rehabilitate the addicts so that they can act like responsible citizens in the community of differentiated cells. So in case somebody's coming in for the first time again, because I know people drift in late, this is the number that you can schedule a personalized health assessment for adjunctive cancer care for you and your loved ones. Okay, so the first thing that I encourage my cancer patients to purchase is a glucometer. Of course, I test their insulin levels, I test their hemoglobin A1C, and so forth. But it's important to understand the relationship of food, exercise, stress levels to fluctuating blood sugar levels. And this is a pretty high level, in fact. If it gets much higher, then it supersedes 180 or so, is where the blood sugar level supersedes the ability of the renal tubules to absorb the sugar and put it back in the bloodstream, and sugar starts spilling out in the urine. Anyway, this is a little lancet kit which has little lancets, so it creates a little poke in the skin. You get a little blood sample, and these guys plug into the glucometer, and you put the little blood spot on the end here, and if it's enough blood within a few seconds, it tells you what your blood sugar levels are. So the first thing I want my cancer patients to do is start measuring their blood sugar levels. The second thing I want them to do is to make sure they don't have any dental problems, mercury fillings or cavitations or root canals, and that'll be the talk of a future webinar, because now we know that's what damages the mitochondria in the body, and that's what drives the toxic injury to the inner cell membrane. They even have little apps. They will have little devices you can attach to your cell phone to measure your blood sugar levels now. They're getting very sophisticated with it. Okay, again, I want to say that a proscription is just as important. For some disorders, it's more important than a prescription. So imagine a prescription pad versus a proscription pad. Proscribe means to forbid, denounce, so forth. So imagine a patient saying, hey, I went to my doctor and they gave me a proscription. And people would say, what? What do you mean a proscription? They gave you a prescription, right? No, they gave me a proscription. They proscribed carbohydrates because I have cancer. They proscribed alcohol because I'm an alcoholic. This is an enormous blind spot that we have in medicine. We don't do much proscribing. Now we could if we put more energy into it. More and more doctors, healthcare providers are being trained in this, but there needs to be a lot more proscribing to balance the prescribing. All right, so how does a family or community deal with addicts and that is in their family or their group? What do they do? Do people say, sure, Mr. Addict, here, consume all the alcohol or heroin or cocaine you want. Here, we'll give you money. You can just take as much drugs as you want. Of course we don't do that. We have all kinds of social treatment, religious and proscribing family values that very few people, unless they're an addict themselves, would ever say that to an addict. So if we don't say that to addicts, then why are people with sugar-addicted sociopathic cancer cells not given the opportunity to restrict their access of their addicted cancer cells to that drug of choice, which is sugar? So again, what I'm saying is, I'm not putting a value judgment here. I'm not saying there's a cure for cancer, but I am saying that whatever treatment a cancer patient engages in, they should be given access to this metabolic treatment, and whatever they use to address their cancer, they almost surely would have to do better adding this adjunctive metabolic approach than they would otherwise. Again, what is a family or community, how they deal with addicts? Do they let addicts run around freely, mugging, lying, cheating, injuring, murdering everyone? Or do they restrict them, lock them up, so that they can't get access to their drugs? Then why are people with sugar-addicted sociopathic cancer cells not given the opportunity to restrain the violent behaviors of their addicted cancer cells? Do they give the addict a chance to rehabilitate and recover to become a productive, normal member of society again? There is evidence that cancer cells can be rehabilitated. So I'm jumping the gun a little bit because this is, again, a future talk that I'm going to give. And I want to also be more complete, more focused on the research. There's a lot of research to read before I make those future presentations. But why then are people with sugar-addicted sociopathic cancer cells not given the opportunity to rehabilitate their undifferentiated cells back into normal, productive, healthy members of the cellular community of differentiated cells? In the human body, there are between 100 and 200 trillion cells in the community. So we offer, does the family or community, we make it clear that they have two choices. You must rehabilitate and get sober or choose to die. And why are people with sugar-addicted sociopathic cancer cells not given the opportunity to force this choice upon their cancer cells, which is choose to become normal, productive, healthy members of the community of differentiated cells, or choose death? Now, everything else you do, I don't care if you inject mistletoe or do hyperbaric oxygen or debulk the tumor with surgery or chemotherapy or radiation, doesn't matter. Whatever works out the best that in your belief system that you think will work, whatever, as you talk about it with your healthcare professionals, whatever you decide to do, please, please, adjunctively include some attention to this metabolic issue. So what is a Paleolithic diet? Paleo diet. How did our ancestors eat for 7 to 10 months a year, depending on your ancestry? Now, you know, if your ancestry comes from Norway, you've got about a 1.5 month growing season or something. So it depends on your ethnicity, but there's a growing season where we had carbohydrates. And then there's a non-growing season where carbohydrates are relatively not available. And because carbs, proteins, and fats all come in the same place, into the Krebs cycle, if the Krebs cycle is working, now that's a big if, say, but if it's working fine, then we do fine without carbohydrates. So, of course, cancer cells can only have carbohydrates, depending on how undifferentiated they are. And we want to impose a carbohydrate sobriety on them by eating mostly low carbohydrate foods. I call this the cancer sober diet. So we can provide this. I provide this for my patients, but we can provide this for individuals that want more information. But here's a list of the vegetables, and there are a lot more vegetables, of course. And here's a list of animal foods. Here's a list of nuts and seeds, not peanuts and cashews because they're too high in carbohydrates. No fruit, many condiments. So just so you don't think I'm blowing smoke, here's an article published in Breast Cancer Research, peer-reviewed paper by Camacho et al. Recent, 2015, Metformin and Breast Cancer, an Evolving Mystery. They think it's a mystery that when you lower blood sugar levels with a diabetic drug, it's a mystery. It's a mystery. How could it be a mystery? With all this evidence, how could it possibly be a mystery? Maybe they had to say that because the editors at this journal would not let them get away with saying metformin and breast cancer, holy smokes, it works. They couldn't say that. So, quote from this paper. In summary, in combination with results from the trials listed in Table 1, the results of Dowling et al. Strongly suggest that metformin exerts a significant part of its anti-tumor effects in breast cancer via the indirect pathway by lowering serum insulin, inhibiting downstream signaling, blah, 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 and leading to decreases in cell proliferation. So as I explored metformin, it originates from compounds derived from the French lilac. The plant has been used since the Middle Ages to relieve the symptoms of diabetes. Isn't that interesting? Numerous early-stage clinical trials are currently underway to determine metformin's potential to prevent an array of cancers and treating them, including colorectal, prostate, endometrial, and breast cancer. A lot of studies are out there. Now they're looking for a magic bullet, say. They're looking for a drug that will treat that. Instead of thinking metabolically, what is actually going on here? But at least they're barking up the right tree. At least we're going to have some data within a few years that will further enhance the validity of the metabolic model in the treatment of cancer. So if you're suffering from symptoms of hypoglycemia, probably a third, a quarter of the listeners know what low blood sugar is: headache, dizziness, irritability, shakiness, and so forth. Can you imagine the suffering of the cancer cells if you had cancer, which are 20 to 100 times more dependent, more addicted on sugar? Can you imagine? They are screaming for mercy. So even lowering blood sugar levels by 5 or 10 or 15 points, maybe blood sugar levels will run 85 normally, say. Even if you can get it down to 70, 65, that will deprive cancer of its addictive support. So this wraps it up, and I'm 4 minutes over, which I also apologize for. We got a little bit of a late start. So cancer is definitely a metabolic disorder. To what extent is it more of a metabolic disorder than a genetic disease? We can't say yet for sure, but the evidence is accumulating that the metabolic angle, the metabolic disorder issue, is very, very important. And when combined with conventional and integrative care, almost has to improve outcomes, the potential for cure, the longevity of any kind of cancer treatment. And you don't have to go very far in the literature to see this. A paleo diet, a diet that takes out the junk food and the processed sugars and junk from the angle of just about any kind of chronic disease, cancer or anything, improves outcomes. So it has to help. So again, this is the number to call for an appointment. I hope that you enjoyed the talk tonight. And if you are interested in being on the email list for continuation of this metabolic discussion of cancer, and we're going to go a lot further than this. This is the beginning. Let somebody know at NIA that you want to that you want that information, and I'll invite you and be happy to have this discussion in the future. I'm wondering if there's any time to ask a few questions on your GoToWebinar panel on the right side. You can type in questions. There may be time for a few. And if you have a proper setup with a speaker and so forth, you can also ask questions, and you can be unmuted. So why don't we open up the possibility to some questions if anybody is interested. I may have explained it so well that nobody has any questions, or perhaps the listeners understood this topic so well that there's nothing new, or perhaps I have put everybody to sleep. So if it's the latter, you can use this webinar, because we're recording it, you can use this webinar to help treat your insomnia. So again, in the right side, there's a panel, and there's a space there where you can type in questions near the bottom if you have any questions. And if you signal, raise your hand to the producer, then they can unmute you. It sounded like someone was trying to come on.
Hi, Dr. Gant, it's Angela.
Hello.
Hi, thank you for your presentation. I was just wondering, for cancer prevention, not active cancer, what blood sugar level do you recommend post-meal, post-prandial, if we are going to use a glucometer to measure our blood sugar for prevention?
It was a little bit garbled. I didn't quite understand. How do we measure it, or what?
I'm sorry. My question is, for cancer